Hi, I'm Will Chou, CEO of Aruvant Sciences. Aruvant Sciences is a privately held company dedicated to developing potentially curative gene therapies for rare diseases. We have two programs. Our lead program is ARU-1801. It's a lentiviral gene therapy for sickle cell disease. We are in an ongoing phase 1/2 trial, and we have clinical data demonstrating a curative potential for this product with up to 100% reduction in vaso-occlusive events. Importantly, ARU-1801 is the only gene therapy or gene editing product in sickle cell disease that has successfully engrafted using only reduced intensity conditioning. Our second program is ARU-2801. It's an AAV gene therapy for hypophosphatasia. This product has the potential to be a one-time therapy that could replace a standard of care enzyme replacement therapy for patients with hypophosphatasia. I'm going to first talk about ARU-1801. So this is a lentiviral product that uses a patient's own stem cells to make a unique fetal hemoglobin. To make our product, we produce our lentiviral vector. We harvest the patient's CD34 positive stem cells. We transduce those stem cells ex vivo. And then ARU-1801 is infused after a single dose of reduced intensity conditioning. Now, the ability to use reduced intensity conditioning has significant benefits for patients, providers, and payers. All of the other gene therapy and gene editing programs currently in investigation for sickle cell disease use myeloablative busulfan. The profile of busulfan is in the center column, and ARU-1801 only requires RIC, reduced intensity melphalan. And you can see there's a dramatically shorter window of neutropenia and thrombocytopenia. This is when patients are at risk of infection and bleeding. No seizure prophylaxis is required for melphalan. The risk of ovarian failure is markedly lower with RIC melphalan compared to myeloablative busulfan. Days in the hospital. So if you get myeloablative busulfan regimen, the inpatient hospital stay is 4 to 6 weeks, whereas in many settings, such as autologous stem cell transplant for multiple myeloma, melphalan reduced intensity conditioning can be done in the outpatient setting. So it is an average of 0 to 5 days in the hospital for these multiple myeloma patients who've received this regimen. So there's a high potential for outpatient administration with RIC melphalan, where there is no potential for outpatient with myeloablative busulfan. Now, the differentiation of ARU-1801 is clearly on the toxicity profile. However, efficacy, of course, is critical for any gene therapy. And the major marker of efficacy is resolution of vaso-occlusive events. And VOEs are episodes of extreme pain that can last several days. 95% of the hospitalizations for sickle cell disease are due to VOEs. They lead to severe complications of progressive organ damage and really to a lower life expectancy. The average life expectancy in the US of patients with sickle cell disease remains in the mid-40s. Now, ARU-1801 has shown significant reduction in VOEs. So each of the red dots you see here is a vaso-occlusive event. And what you see is the 24 months pre-treatment with ARU-1801 compared to the 24 months post-treatment. The first two patients were treated with a academically manufactured ARU-1801, and they had 93 and 80% reduction of VOEs, so a dramatic improvement. Patients 3 and 4 were the first patients treated with a improved manufacturing process since Aruvant acquired the product, and those patients have had no VOEs to date. Now, there are some unique attributes of ARU-1801 that allow this product to have a therapeutic effect with a lower product vector copy number, and also that allow this product to engraft with less conditioning. So it requires less stem cell clearance. The first unique factor is ARU-1801 has a proprietary point mutation, the G16D point mutation, that drives higher hemoglobin F payload per vector copy. The second effect is that our anti-sickling hemoglobin payload, hemoglobin F G16D, itself has a more potent anti-sickling effect than endogenous hemoglobin F. And third, we have a proprietary manufacturing component that we add to our manufacturing process that enables us to transduce and transplant more true stem cells. So I'm going to take each of these unique features one at a time. The first is that the proprietary G16D point mutation drives a higher anti-sickling payload per vector copy. And we first showed this in preclinical studies. The mouse models of sickle cell disease on the left compare a mouse who was transplanted with a wild type gamma globin compared to a mouse who was transplanted with a G16D gamma globin. And what you can see is that per vector copy number, the mouse that had the vector that transduced a G16D gamma globin had a 1.5 to 2 times more hemoglobin F payload expression compared to the mouse that got an endogenous unmodified gamma globin. So this G16D point mutation in itself enables more hemoglobin F formation per vector copy. And so that was preclinical models. And what we are seeing in our first patients who we've treated is something very similar. So if you look at the same metric, payload per drug product BCN, our payload, hemoglobin F G16D per drug product BCN, is about 30%. And the total anti-sickling hemoglobin is about 45% per unit of drug product vector copy number. This is markedly higher than what is seen in other lentiviral therapy, lentiviral gene therapy programs in sickle cell disease. And what this means is that we can achieve therapeutic anti-sickling hemoglobin levels with lower product vector copy numbers. The second effect, and this is some of our data from ASH recently, is that hemoglobin F G16D appears to have a more potent anti-sickling effect than endogenous hemoglobin F. And patient 4 is a great natural experiment. What you see here for patient 4 is in blue, their levels of endogenous hemoglobin F, so unmodified hemoglobin F, and in orange is their hemoglobin F G16D. And this first bar is at screening. So before treatment, this patient was on hydroxyurea and had a level of hemoglobin F of about 15%. After treatment with ARU-1801, they're of course taken off hydroxyurea. And at about 12 months after treatment, they have about 15% hemoglobin F G16D and a very small amount of endogenous hemoglobin F. What you can see is that even though they have very similar levels at screening and at 12 months of total hemoglobin F, most of the hemoglobin F now is hemoglobin F G16D, and now they have a markedly different phenotype as well. They have 0 VOEs in these 12 months versus 9 in the prior 12 months. And we see this not only in the phenotype of VOE, but we also see this in the actual functioning of the red blood cells. So the data on the right is ectocytometry. And the way ectocytometry works is you take a patient's red blood cells, and in this machine, you slowly decrease the partial pressure of oxygen. So you move from the right to the left. The y-axis is a measure of stiffness of the red blood cells. So when the red blood cells get stiff, that is when they sickle. And so at baseline, this patient's point of sickling, the partial pressure of oxygen when they sickle, as you move down here, this is the point of sickling. The point of sickling at baseline was 51, and now 12 months after ARU-1801, the point of sickling has now shifted down to 30, which means if the partial pressure of oxygen in venous tissues is about 40 millimeters of mercury, and so that means at 30, by getting the point of sickling down to 30, this is below the threshold of where oxygen gets in venous tissues. This is why the patient isn't sickling. So we see a change not only in the ultimate phenotype we care about, VOEs, but we also see the change in the actual functioning of the red blood cells. And the very interesting point is our third patient had an even higher level of hemoglobin F and hemoglobin F G16D. They had 38% total hemoglobin F. And that patient is shown on ectocytometry in yellow here. And at 12 months, patient 3 with a 38% total hemoglobin F, their point of sickling is 27. So it looks very similar to patient 4's. So it appears we may have we may be seeing in patient 4 a bit of a threshold effect that if we get hemoglobin F G16D levels to about 15%, we can dramatically improve the phenotype of sickle cell disease. Now, finally, we have a unique additive to our manufacturing process that keeps stem cells in their stem state. So when you put stem cells into culture, they want to differentiate into progenitor cells. Progenitor cells are more differentiated and don't live forever. Therefore, they don't contribute to long-term engraftment. So if you're transplanting transduced progenitor cells, they don't really give any durable benefit over a long period of time to the patient. So this stemness enhancer inhibits stem cells from differentiating in culture when we're manufacturing the product. Therefore, we transduce and transplant more true stem cells. So for any given product vector copy number, there's a higher chance of engraftment and long-term durability. Now, does using reduced intensity conditioning matter to doctors? Well, from the market research we've done with hematologists and transplanters, it does really matter. So what we asked both hematologists and transplanters is, of your patients with severe sickle cell disease, how many would you consider eligible for gene therapy? And we first asked them in the setting of if there was only a myeloablative gene therapy or gene editing product available on the market. And they said 49% of their patients would be eligible. And now we said, suppose now there's a myeloablative option, but also ARU-1801, which uses only reduced intensity conditioning, available. They then said about 70% of these severe sickle cell patients would be eligible for gene therapy. And of those patients who ultimately do go get gene therapy, as you see, some patients who are eligible still don't receive gene therapy for a variety of reasons. But of those who actually get gene therapy, the majority would receive the RIC option, which would be ARU-1801. Now, ARU-2801 is a one-time AAV gene therapy for hypophosphatasia. It could replace a blockbuster enzyme replacement therapy standard of care, and it delivers potentially curative efficacy without the current limitations of chronic administration that the current ERT has. So it's shown durable increases in the missing enzyme, tissue nonspecific alkaline phosphatase, through 18 months, with a potential for a significant reduction in disease burden. So first, what is hypophosphatasia? It's a devastating, potentially fatal orphan disorder with no gene therapy treatments available. It's caused by a mutation in the ALPL gene, and it's characterized by a low or non-existent expression of tissue nonspecific alkaline phosphatase. And without TNS-ALP, really, these patients do not mineralize bone. So their bones are too soft. That is the main issue with HPP, but it can lead to many severe complications. The most severe forms result in respiratory failure, seizures, limb deformity, and left untreated, half the patients with the most severe type of HPP die within the first year of life. Now, Strensiq is the current enzyme replacement therapy that is available for HPP, and it has dramatically improved the life of patients with HPP. It's made a huge difference to the patient population, and really, it's been a very successful launch. So sales are expected to exceed $1 billion. So we know this is an established high potential market. There's a regulatory and development path that has been informed in the past by Strensiq. However, Strensiq has some issues of its own. So it requires up to 6 subcutaneous injections a week for a lifetime. And frequently, patients get adverse events at the injection sites. They get lipodystrophy and other injection site reactions. And what patients have told us is, I wouldn't hesitate with something new if it meant less injections. And another patient said, every injection is the most dreaded experience. It's like asking to inject fire into your body. So a one-time therapy that could replace 3 to 6 subcutaneous injections a week has a huge unmet need from the patient population. And of course, there was a huge unmet need from a payer standpoint as well, to not have a lifetime of therapy. And finally, this is some of the preclinical data that has gotten us so excited about ARU-2801. And so what you're looking at here is a HPP murine model. These are and so in these murine models of hypophosphatasia, the y-axis is ALP activity and the x-axis is months. And you can see in the lower left here, this is an HPP mouse with no treatment, and you can see the survival is very low. So this is the most severe phenotype of HPP, and it's less than 1 month survival. The dashed line is a wild type mouse. And then these top lines are HPP mice who are treated with different doses of ARU-2801. And what you can see is a durable increase in ALP levels out to 18 months. And in the higher dose, which is 1E12 per mouse, you can see that this higher dose has an overall survival of 70% at 18 months, and that is the duration of the study. Importantly, at these high levels of ALP, there was no macroscopic or microscopic evidence of ectopic calcifications. That would be the primary side effect specific to this product that one would worry about. Also, at the doses that we are looking at, they are at least a log lower than doses that have encountered any toxicities in the past when we talk about problems with other AAV therapies. So in summary, ARU-2801 has a product profile that's positioned to lead the market. It eliminates chronic administration. It's shown durable ALP increases, and it has a generous therapeutic window. The path forward for us is that we're going to have multiple clinical and preclinical milestones in 2022. We will complete our enrollment of our phase 1/2 trial. We will be updating mid-year and at the end of the year on the progress of ARU-1801. We will have new data from ARU-2801 non-clinical studies, and we'll be initiating our pivotal trial in 2023. Thank you very much for your time, and I look forward to further conversation.